Okay, next is about sclerenchyma tissue. Okay, so you see the structure. What are the main difference? What are the main difference between parenchyma and collenchyma? So if you see here, normally you see here, the main difference is it does not contain nucleus. Tak ada nucleus punya. And then also tak ada nucleus and also no protoplasm. No protoplasm. And then this center actually is not vacuum. It is not a vacuum. It is the empty lumen. It has empty lumen. Yang ni adalah hollow punya. Memang empty punya. Tak ada apa-apa pun. Ini bukan vacuum. Ini memang tak ada apa-apa. So normally if you see that it has thick lignin cell secondary cell wall, most dead cell. So it is dead cell, very major and no protoplasm. So that is why it does not contain nucleus and chloroplast one, because it is the dead cell one, dead cell. So it does not contain organelles lah. Tak ada organelle yang boleh fungsi punya. So that is why we call as dead cell. And it is very tightly packed with each other with no intercellular space. So langsung tak ada space punya between the cells. Compared to collenchyma still has some small cell space, but for sclerenchyma does not contain any space and it has empty lumen lah. So what are the main functions? So the main function of the sclerenchyma normally is provide mechanical strain, provide mechanical support, because it is the dead cell. It does not contain nucleus and chloroplast, so it cannot carry out photosynthesis one. So normally it just provide the support and strain only, and then it maintain the rigidity of the plants, and also can exchange material between the adjacent cell. So although it is dead cell, but the transport between the nutrient still can occur. Still it can occur because it contains cell wall and plasma membrane, so it still can function. Just that it does not contain more organelles lah. Okay, so normally sclerenchyma tissue it contain two types. Either is in longitudinal, either in fiber or the fiber one, or the yang ni call trachea. Fiber or trachea. Yang ni fiber, yang panjang-panjang punya longitudinal cross section is the fiber. This one that you see here is we call as trachea. Okay, so what are the function of the fiber? Not trachea, scleroid. Sorry. Okay, so fiber structure. So you see that the fiber structure is more long and standard cells occur in group in strand, and then fiber is also part of the xylem and phloem tissue. So this one later we will learn vascular tissue. So actually fiber tissue is also the part of the xylem and phloem tissue, and then it has the fewer peaks compared to the scleroid in the cell wall. So the peak of the fiber is more or less the number of the peak is lesser than the scleroid cell, and then found in the leaf of the flowering plant. So normally fiber function is to provide mechanical support and strength for vascular tissue. Okay, so this one is the fiber. You will see that under the microscope. So it is more elongated shape one. Okay, so next is about scleroid. So scleroid is the spherical shape, and most may live or dead at maturity. Normally is dead and consists more peat than fibers in the cell wall and found in shell or nut and stone of the fruit. So normally this function is to protect the seed from destruction, give gritty texture structure to peel. So this one this is Chinese lah. In Malay we call as kasar lah, more gritty texture, meaning that it is more kasar punya surface, not smooth and shining one surface, more kasar punya surface lah. And also make the cell more harder and inflexible. So these are the main function. So this function that scleroid cell does not provide mechanical support one. It only provide to become the cell more harder. Okay. Okay, so this is the structure under microscope. So you see that the function is to strain scary-rich tissue is hard and inflexible. Okay. Okay, so that's all for ground tissue. So this all of these you should know all of this structure because maybe in examination, especially in essay question, it maybe want you to draw it. So if you want to draw it, so you need to know lah. What are the shape? Either it has the thick cell wall or thin cell wall, or it has the space or not. All of this you need